Hello A&P1 students, this is Gina Piscatelli with a final lecture on neuron physiology. This is for week 7. In this lecture, we'll focus specifically on synapses, that is how a neuron communicates with target cells. And sometimes neurotransmitters are used in order to communicate. These are chemicals that the neuron releases and a target cell can bind to if it has a receptor for that specific neurotransmitter. So that's our topic for today, and we're going to start off by looking at what is the definition of this word synapse. Synapse is a junction between two cells, so we call it an intercellular junction, and it exists between a neuron and then a target cell. And there are 3 types of target cells: a muscle cell, another neuron, or a cell in a gland. And I have pictures on this slide of a neuron innervating or forming a junction with a muscle cell. That's the picture with the green neuron and the red muscle cells, and you can see some nice axon terminals branching out around the muscle cell. On the bottom left picture, there's more synapses on another neuron. So the neuron is the target cell, and this is the cell body of a neuron, and many axon terminals and synaptic knobs are forming junctions with this cell body. And of course, those axon terminals are from a different neuron. So we often use, when we're talking about 2 neurons forming a synapse, we often use these terms presynaptic and postsynaptic. And you've seen this before, but I just wanted to remind you that a presynaptic cell is the cell that's sending a signal to a postsynaptic cell, the cell that receives the signal. And the junction, the place where they meet, is called the synapse. Now, sometimes the axon terminal will form a synapse on a dendrite. Other times, an axon terminal will form a synapse right on the cell body of the postsynaptic cell. So there's different types of synapses. Sometimes a synapse acts by an electrical signal flowing from the presynaptic to the postsynaptic cell. This is very quick because really all it is the flow of ions from one neuron to the next neuron. And other times a synapse is what we call a chemical synapse. This is slower. The signal is a chemical known as a neurotransmitter released by the presynaptic cell. And then that neurotransmitter binds to a receptor on the postsynaptic cell. So here's some pictures of electrical synapses versus chemical synapses. So the picture on the top is a electrical synapse, and what you can see is the 2 membranes, presynaptic and postsynaptic, are joined or adhered by these channel-like structures. These are called gap junctions, and they allow a certain ion to flow from a presynaptic neuron to a postsynaptic neuron. But I know I've drawn it from top to bottom, but it could just as easily go the other way if the cell on the bottom started sending a nerve impulse first. So there's some flexibility with this electrical synapse. You find electrical synapses in parts of the brain particularly, and the spinal cord, but mainly the brain. Chemical synapses are very common. They include a neurotransmitter diffusing across a space that exists between the presynaptic neuron and the postsynaptic neuron. This space is called the synaptic cleft. And so a neurotransmitter is released by exocytosis from the presynaptic neuron, and it diffuses across the synaptic cleft and binds to a receptor on the postsynaptic cell. This is very common in muscle, glands, and parts of the nervous system. Now, there are advantages to an electrical synapse. It's really fast, and the current or the signal could flow in either direction. It depends on which of these neurons fire a signal first or send a nerve impulse first. So you have to really name pre versus post-synaptic neuron based on the direction of the current or the signal. Now, the disadvantage is that it's always excitatory. There's no way the signal is ever inhibitory because depolarization is a positive charge, and that positive charge gets transmitted into the next cell, postsynaptic, and that depolarizes the postsynaptic neuron, leading to an action potential, which is excitatory. So there's not very much versatility in terms of inhibiting a neuron, but it's very, very quick, and it can go in two directions. So let's look at a chemical synapse one more time. So here we have the structure anyway. Here we have a presynaptic cell, or neuron, and an axon terminal. The depolarization comes down to the axon terminal, which causes the secretion, or by exocytosis, of a neurotransmitter, shown in kind of aqua blue. And then that neurotransmitter diffuses across the cleft, and it binds to a receptor on the postsynaptic cell. Now that will open up a channel, an ion channel, frequently. This isn't the only thing a neurotransmitter can do, but it's one of the things. So we call that receptor a ligand-gated ion channel because it allows an ion to then flow into the postsynaptic cell. So in this way, it's not different than the electrical synapse. You're still talking about ions getting into a postsynaptic cell, but there is quite a delay. And something else to mention is that these receptors on postsynaptic cells vary quite a bit and have different actions, which we'll talk about towards the end. So this picture I just thought was fun to look at. It is a picture of an axon terminal at a chemical synapse. What's shown in blue are vesicles that contain neurotransmitter. So what they did was they broke a synapse. They took an axon terminal off and took a scanning electron microscope photograph and then colored it. So this is not the way your axon terminals look. They don't look orange, blue, and green. But I just thought it was really cool. Okay, so this delay in the chemical synapse is referred to as a synaptic delay. So you get depolarization coming down, and then it takes some time for diffusion across the synaptic cleft. We say the synaptic delay is usually about 0.5 milliseconds. Now, in order for exocytosis of the neurotransmitter to occur, calcium influx is required. So what happens is that depolarization or action potentials spread down to the axon terminal. This is all normal, nothing new. Once the depolarization reaches the axon terminal, certain calcium channels are opened. They are localized at the axon terminal. They're green in this picture. And we call them voltage gated because they open due to depolarization or positive charge. When they open, extracellular calcium flows into the axon terminal. That calcium is needed for synaptic vesicles to fuse with the presynaptic membrane. So for those secretory vesicles to fuse right here, you need calcium. Now the neurotransmitter is released, it diffuses across the cleft and binds to whatever receptor is on the postsynaptic cell for that particular neurotransmitter. Now the advantages and disadvantages of a chemical synapse exist just like we had with the electrical synapses. So one disadvantage is that it's slow. It's much slower. But the advantages outweigh the disadvantages. For example, a presynaptic neuron can vary how much neurotransmitter is released. So the more depolarization or the more impulses, the more you're going to get neurotransmitter released, and then you'll have more effect on the postsynaptic neuron. In addition, the postsynaptic neuron can change its ability to respond to neurotransmitter. Now, it's not like it thinks about this, but biologically what can happen is a postsynaptic neuron can change the number of receptors it has. It could have more receptors or less receptors, right? So more receptors, you would get more of an effect. Less receptors, you'd get less of an effect. Another big advantage not seen in an electrical synapse is that the postsynaptic neuron might be excited by a neurotransmitter or inhibited by a neurotransmitter. Now, this depends in part on which neurotransmitter we're talking about. Okay, I'm just going to abbreviate neurotransmitter NT. But even a single neurotransmitter, like acetylcholine, can have different effects on different target cells because postsynaptic target cells can have different receptors. So the type of receptor that a postsynaptic cell has is what's going to determine whether a neurotransmitter is truly excitatory or inhibitory to it. So we're going to look at some examples of those so that it makes more sense to you. Right now it's just theory, right? So the concept of a synapse being either excitatory or inhibitory. Let's talk about this. So an excitatory synapse means that the postsynaptic cell is going to experience depolarization. An inhibitory synapse means that the postsynaptic cell won't experience depolarization, but even more than that, it will experience the opposite, hyperpolarization. It will become more negative on the inside. Excitatory, positive, inhibitory, negative. Okay, so here's some examples of synapses. Named synapses. Cholinergic synapse is a synapse in which the presynaptic neuron releases the neurotransmitter acetylcholine. An adrenergic synapse is an example of when the presynaptic neuron releases adrenaline, also called epinephrine. And a GABAergic synapse is a synapse in which the presynaptic neuron releases this thing called GABA, which is gamma-aminobutyric acid, which you will never have to remember. Just GABA is fine. Okay? Now, what are the effects of the neurotransmitter for each of these synapse types on the postsynaptic cell? What does a cholinergic synapse do to the target cell? What does an adrenergic synapse do to the target cell? And what does a GABAergic synapse do to the target cell? So let's look at them specifically. And I'm going to split them up into examples that are excitatory to a target cell and inhibitory to a target cell. Acetylcholine is a neurotransmitter that is excitatory to skeletal myocytes. So this would be an example of an excitatory cholinergic synapse. Norepinephrine or adrenaline or epinephrine can also be an excitatory neurotransmitter, and it is to cardiac muscle cells. So you know your heart rate goes up in response to adrenaline, right? So adrenaline is excitatory to cardiac cells. Now, some examples that cause inhibition of a target. Again, I chose generally muscle. So, interestingly enough, the neurotransmitter acetylcholine actually inhibits cardiac muscle contraction. So it does excitation to one kind of muscle, but inhibition to a different kind of muscle, and we're going to look at why. GABA is always an inhibitory neurotransmitter. There are some neurons in the central nervous system that respond to GABA and are inhibited. This is important to make sure that your brain is focusing on what it needs to focus on at any given time. So let's look at how an inhibitory synapse might work, because we haven't talked about an inhibitory synapse at all. GABA is the major inhibitory neurotransmitter in the brain. And so neurons, which we'll call presynaptic neurons in the brain, release this neurotransmitter GABA that binds to postsynaptic neurons in the brain. Those are usually the targets. And the receptor on the postsynaptic cell, this orange structure, is a chloride ion channel. So when GABA binds to the receptor, that channel opens. Chloride ion diffuses into the postsynaptic cell. Well, that's a negative charge. So the postsynaptic cell has been hyperpolarized and is less likely to achieve that very first action potential that's needed for a nerve impulse. We've already looked at one example of an excitatory synapse. I'm going to just give the neurotransmitter a name now. The neurotransmitter is acetylcholine. When it's released to the target cell of skeletal muscle. So we call this a cholinergic synapse. Acetylcholine diffuses across the cleft from the presynaptic neuron to the postsynaptic neuron, or muscle, skeletal muscle, postsynaptic muscle cell. And that acetylcholine binds to a receptor that is a sodium channel. So now sodium flows into the postsynaptic cell. The inside of the cell becomes positive. It's depolarized. And so you'll get an impulse in the postsynaptic cell, which results in muscle contraction. So your skeletal muscles will contract in response to this excitatory neurotransmitter. But now we want to know, how can acetylcholine be excitatory in one example, one cell, skeletal muscle, and then also inhibitory, but in a different type of target cell, the cardiac muscle cell. It's because there are different receptors for acetylcholine, different cholinergic receptors located in different target cells. The two types of receptors, which you don't really have to remember the names, are nicotinic acetylcholine receptors and muscarinic acetylcholine receptors. The nicotinic receptors are in skeletal muscle, and the muscarinic receptors are in cardiac muscle cells. So we know that acetylcholine is excitatory to skeletal muscle cells. We know that. So it must work in some way by getting sodium into that postsynaptic cell. And sure enough, it does. It's very much like what we've already seen. The postsynaptic cell, postsynaptic cell has a sodium channel. So sodium goes in. I can't get this thing to work right. Sodium goes into the muscle cell. The muscle cell is depolarized and contraction occurs. Now, muscarinic receptors don't work like the GABA inhibitory ones. They are inhibitory in cardiac muscle cells, but a different mechanism is used. Acetylcholine from the neuron binds to a receptor, a muscarinic receptor, on cardiac muscle cells, and this receptor is linked to a G protein. So it's shown in purple in the diagram. This is the receptor. Acetylcholine is the little round purple ball. And notice that there's a G protein or two kind of attached, or at least close to the receptor. When acetylcholine binds to that receptor, the G protein is activated and it affects a nearby enzyme, which you do not have to remember the name of, called adenylate cyclase, which normally produces cyclic AMP. But the G protein inhibits that. So that means cyclic AMP goes down. And when you have low cyclic AMP, evidently these calcium channels close. So calcium is not going to be able to get into the muscle cell. And you're not aware of this yet, but we need to have calcium in a muscle for contraction to take place. So when you have low calcium, you have decreased force and rate of heart contraction. Now, I'm not expecting you to remember the names of these receptors or specifically how this particular muscarinic acetylcholine receptor works. It's just used as an example to show you that this idea of a G protein linked receptor can cause different effects in a postsynaptic cell using the same neurotransmitter. So acetylcholine can be excitatory or inhibitory. Okay. And then finally, we should talk about how does the signal ever stop? How does it ever, the postsynaptic cell, ever not, ever become quiet again, not stimulated anymore? Well, first, I'm sure neurotransmitter isn't released anymore by the presynaptic neuron. So that's one thing. It's just not, there's no more stimulation. But yet, these receptors on the postsynaptic cell have been activated. They have bound to neurotransmitter. So there's got to be a mechanism for stopping the process. And what happens is that with a certain rate, the neurotransmitter will actually come off of the receptor, because everything's a chemical reaction. So binding will stop, and then the neurotransmitter just diffuses out of the synaptic cleft. It just goes away. And that's what number 1 shows down here, is that the neurotransmitter has just kind of come off and diffused away. But in some cases, a synaptic cleft has an enzyme that breaks down the neurotransmitter. Acetylcholine is one example where we know the name of the enzyme that breaks it down. It's called acetylcholine esterase, no surprise. So that's what's happening in number 2, is the neurotransmitter is being degraded. But there's another phenomenon that takes place, and that is that the neurotransmitter can be recycled. So the neurotransmitter comes off of the receptor, and often there are transport proteins that help the presynaptic neuron take the neurotransmitter back up into its cytoplasm. We call these reuptake transporters. Okay, sometimes they're in the cleft and sometimes they're in the membrane. So the neurotransmitter can be recycled. It can be used again by the presynaptic neuron. That's everything for synapses and neurotransmitters. Thank you very much for your attention.